Hi, in this module and the next one, I'll talk about the pre-processing of fMRI data. So if you recall our data pre-processing pipeline, we first talked about data acquisition and reconstruction, and then we talked about experimental design. Now we're moving on to talking about pre-processing, which is a necessary step before we can talk about data analysis. So prior to analysis, fMRI data undergoes a series of pre-processing steps, which are basically aimed at identifying and removing artifacts and also validating certain model assumptions. In general, there's a threefold goal with pre-processing. One, we want to minimize the influence of data acquisition and physiological artifacts. Two, we want to check statistical assumptions and transform the data to meet these assumptions. Three, we want to standardize the location of brain regions across different subjects to achieve validity and sensitivity in group analysis. Now, pre-processing is performed both on the fMRI data and on the structural scans that are collected prior to the experiment. So basically, there's an interplay between the pre-processing performed on the structural scans and the functional image time series. And this is a cartoon of the pre-processing pipeline that we often use. And we'll return to this once we've explained all the different steps that we're going to go through. So there are a number of different pre-processing steps, and they include visualization and artifact removal, slice time correction, motion correction, correction for physiological effects, co-registration and normalization, and spatial and temporal filtering. So let's start with talking about visualization and artifact remover. So that's always should be the first part of any pre-processing pipeline. And here we use exploratory data techniques to investigate the raw image data and detect possible problems in artifact. So you'll recall from a previous module where we talked about the different artifacts that can appear in fMRI data, we want to ensure that none of these are causing any problems in our data. So, for example, fMRI data often contain transient spike artifacts or slow drift over time, and we want to be able to control for this. So, any exploratory techniques such as principal components can be used to look for these spike-related artifacts. Here we see an example of a few spike artifacts that we want to somehow take into consideration in our data analysis. We'll talk about how to do this in subsequent modules. We often sample multiple slices of the brain during each individual repetition time to construct a brain volume. However, each slice is typically sampled at slightly different time points because we acquire them sequentially. And so even though we're treating a brain volume as being scanned at a single time point, this is usually not the case. So the top of the brain volume might be sampled maybe a second or two later than the bottom of it. And this is something that we need to correct for. So slice time correction shifts each voxel's time series so they appear to have been sampled simultaneously. So let's illustrate this. Let's say that we have 3 slices of the brain, and in each of we have a voxel from 3 different slices, and they all have the same hemodynamic response signal in them. However, because of the way that we're sampling the data, we'll sample the green data points in slice 1 every second, and the yellow at the yellow time points we'll sample the voxel in slice 2, and it's going to be shifted vis-a-vis slice 1, and the same for slice 3. So if we extract a time course from slice 1, slice 2, and slice 3, and we don't correct for slice time correction, they're going to look quite different even though they come from the same underlying signal. And so this is something that we want to guard against and kind of correct for. So this can be corrected using temporal interpolation and other methods. So temporal interpolation, we use information from nearby time points to estimate the amplitude of the MR signal at the onset of each TR. And here we can use linear, spline, or sinc functions to interpolate it. Alternatively, we can use Fourier methods and phase shift methods and slide the time course by applying a phase shift to the Fourier transform of the time course. Either of those methods work fine. Head motion is a serious problem in neuroimaging, and even very small movements of the head during an experiment can be a major source of error if not treated correctly. So when analyzing a time series associated with a single voxel, we always assume that it depicts the same region of the brain at every time point. However, if we move our head around, one voxel that was in this position might be in a completely different brain area because of head motion. And this is something that we want to guard against. And so this can usually be corrected using something called rigid body transformation. Here we assume that the brain is a rigid body that might move around, and we try to correct for this movement. And in general, we do this, and the goal here is to find the best possible alignment between an input image and some target image. So to align the two images, one of them needs to be transformed so that it lies on top of the other image. And here we use what's called a rigid body transformation. And a rigid body transformation involves 6 variable parameters. We have 3 sets of translations in the X, Y, and Z direction, and 3 sets of rotations, pitch, yaw, and roll. Here's an example of a translation. We might translate this guy in the XY direction. Here's an example of a rotation. We might rotate the image in a direction like that. And here's scaling is another type of transformation that we might do. We might scale the image. And shearing, we might shear the image as well. So in general, these are different types of linear transformations that are often used in neuroimaging. So the rigid body transformation that we use in motion correction involves 3 translations and 3 rotations. A similarity transformation involves the same 3 translations and rotations and also a single global scaling function. Finally, an affine transformation includes 12 degrees of freedom. We have 3 translations, 3 rotations, 3 scaling parameters, and 3 shearing parameters. There's also warping methods where the transformations, where the equations relating the coordinates of the images are nonlinear, but we're not going to talk about those in detail in this class. So in motion correction, we do a rigid body transformation with 6 degrees of freedom. And here the target image is usually defined to be the first or sometimes the mean image in the fMRI time series. But let's just say, for the sake of argument, it's the first one. And now what we want to do is we want to align all subsequent images to that first image to make sure that the voxels are aligned with one another. And so here the goal is to find the set of parameters which minimizes some cost function that assesses the similarity between the image and the target. So let's say that we're looking at the second image now. We want to say, well, how can we assess how similar, how much overlap there is between the first and the second image? And so examples of different cost functions can include the sum of the square differences or the mutual information. So here's sort of a cartoon here. So we have a target brain here, and we have another brain which we want to align to it. So basically we have to find a set of rotations and translations so that this blue brain is overlaid onto the green brain. And so that's the goal of motion correction. And so here is the results of a motion correction. And again, we have 3 translations and 3 rotations. And so this is showing the translations in the X, Y, and Z direction for each image when we're trying to align it to the first image, and also the 3 rotations, pitch, roll, and yaw. After we do motion correction, a structural MRI is collected in the beginning of the session, is registered to the fMRI images in a process known as co-registration. So even though the structural image and the functional images are acquired during the same scanning session, they're often not completely aligned with each other, partly because they're measuring different things, but partly because people may have moved throughout the session. And so by co-registering the structural and the functional images to one another, this allows one to visualize single subject task activations overlaid on the individual's anatomical information. So this is sometimes a nice way of presenting results. So even though the statistical analysis is on low resolution functional data, we might want to present the results of the analysis on a high resolution structural scan where we can make out detail. It also simplifies later transformation of the fMRI images to a standard coordinate system, which we'll talk about later. And so there's certain key differences between co-registration and motion correction. Here, the functional and structural images don't have the same signal intensity in the same areas because they have different contrast. So they can't be directly subtracted to each other. Also, their shapes may differ from each other. So here we have to use at least an affine transformation with 12 degrees of freedom to perform co-registration, and often a mutual information cost function is used. Here we see the example of a co-registration in the software program SBM, where the functional images has been co-registered using mutual information onto a structural scan. Okay, that's the end of this module where we've gone over a couple of things in pre-processing. In the next module, we'll continue the pre-processing pipeline and talk about normalization and spatial smoothing. Okay, see you then.